In this topic, we're going to look at the introduction to mutations and gene mutations. So by the end of this topic, you should be able to answer these questions. What is a mutation? What environmental factors increase mutations? What's a gene mutation? What are the different gene mutations that you get? What's a frameshift? What is sickle cell anemia? And then finally, we're going to look at phenylketonuria. So what I want you to do is press pause, copy down these questions so that you can use them for revision later. Okay, just to recap on what a gene is. It's a section of DNA that codes for a particular protein. The position where it's found on the chromosome is called the locus. So here you can see the different colored regions represent the genes. And a gene is made up of a sequence of nucleotides, each with its own base. Can you remember what an allele is? It's a different variation of a gene. For example, you've got an allele for blue eyes or brown eyes. And the different alleles of a gene contain slightly different sequences of bases. Okay, let's move on to what is a mutation. Any change to the quantity or the structure of the DNA of an organism is known as mutation. It's a spontaneous, unpredictable change in the genetic material. So mutations that occur in the body cells will not be passed on to the next generation, but those that occur during the formation of the gametes may be inherited, and they often produce sudden and distinct differences between individuals. So you can get a mutation where the number or the structure of the chromosomes is changed, and this is called a chromosome mutation. It's caused by errors that occur during cell division. If there's a change to the DNA that affects a single locus and produces a different allele of a gene, we call this a gene mutation. So this is when the DNA is changed, so you get a different allele being produced. And it's caused by errors that occur during DNA replication. So mutations may occur completely randomly with no obvious cause. However, you do get several environmental factors that increase the change of mutation occurring. Can you think of a few? Well, you've got ionizing radiation, for example, alpha, beta, and gamma, and these can change DNA molecules, altering the structures of the bases within them. Ultraviolet radiation, it's got a similar effect to ionizing radiation. And then chemical mutagens, for example, mustard gas. So a substance that increases the chance of mutation is called a mutagen. Okay, gene mutations. These are also called point mutations. And these are changes in the DNA structure. So it's going to change the mRNA that reads the DNA. This will also result in a change in the amino acid sequence. So this means that you might get a completely different protein being coded for. Okay, you've got different gene mutations, and your textbook describes 3 different ones. You've got addition, deletion, and substitution. We're also going to look at duplication and inversion. Okay, here's an example of addition. As the name suggests, this is when one or more extra bases are added to the sequence. So my alphabetical letters here represent the nucleotides. Okay, deletion, this is when one or more bases are lost from the DNA sequence. Substitution, the nucleotide base is substituted for another base. As you can see here, the letter E has replaced the letter C. Duplication, this is when a section of DNA has been duplicated. And then inversion, this is when a section of DNA has been inverted, so the base sequences appear to be swapped around. Okay, let's have a look at a frameshift. Well, addition, deletion, and duplication will alter the way the triplets of bases are read, and thus the sequence of amino acids that make up the polypeptide will change. For example, you've got the DNA code AGT, AGT, AGT. It's going to code for the amino acids serine, serine, serine. Now, if you mutate that DNA sequence by addition, deletion, or duplication, the amino acids they code for are going to be changed. This means that the amino acid sequence is also going to be changed. So the result is called a frameshift if the DNA code is changed so that every triplet of bases that follows the change is altered. So the new protein can be completely different. So either it's useless or you get an incomplete protein. So if you get an incomplete protein, this is when the triplet code has coded for a stop codon. Okay, let's have a look at addition. So what happens during addition? If you add a base, for example cytosine, into the base sequence, have a look at what's going to happen to the DNA sequence. Can you see that it's shifted to the right? Now have a look at the amino acids that are coded for. Are they the same? No. So this shift in the right is going to affect the codons, and it's also going to affect the amino acid sequence. So you're going to get a protein with a completely different sequence just from the addition of one nucleotide. So instead of you getting the amino acid serine, 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 you're going to have cysteine, isoleucine, isoleucine. What do you think is going to happen if you duplicate the nucleotide A? It's going to have a similar effect to addition. So you can see this frame shift to the right. This means you've got different amino acids being coded for. Do you remember how to look up the amino acids? If you have that table of codons and amino acids, have a look at either, if you have a DNA versus amino acid table, have a look at what the DNA sequence, the triplet code codes for, or the codon mRNA code for. If we delete guanine, what do you think is going to happen? There's going to be a frame shift so that you get different amino acids being coded for. So notice the difference in just one deletion, how it changes the whole sequence of amino acids because of the sequence of nucleotides. Okay, if you have substitution, so if a nucleotide in a DNA sequence is replaced by another that's got a different base, this type of mutation is called a substitution. Now, if it doesn't have any effect on the amino acids coded for, as you can see here, serine is still coded for, even though you've changed the base from thymine to adenine, we call this a silent mutation. So what's the definition of a silent mutation? This is when you've got a substitution that results in a different base occurring in a DNA triplet. For example, T has been replaced by A. But one that still codes for the same amino acid. So even though you've substituted one nucleotide, you've still got the same amino acid being coded for. So silent mutations have a final polypeptide is going to be identical to the original and no effects are apparent. Now you do get some substitutions that will result in a different phenotype. An example is the inherited disorder sickle cell anemia. So this is the result of a gene or point mutation, more specifically a substitution, in the gene that produces hemoglobin. So the result is that the red blood cells are going to be pulled out of shape into this half moon shape, as you can see here. And this is what we call a sickle cell shape. So if you remember, hemoglobin molecule is made up of 4 polypeptides, and each one's got a heme group. So you've got 2 alpha chains and 2 beta chains. And the gene that codes for the amino acid sequence in the beta chains is not going to be the same in everyone. So most people, this is the sequence of amino acids. Now I want you to have a look at what happens to amino acid 6 in people who've got sickle cell anemia. So in amino acid 6, glutamine is going to be changed to valine. This is what causes sickle cell anemia. So this change in one amino acid is going to have a serious effect on the hemoglobin molecule when the molecule is not bound to oxygen. Okay, you don't need to know this in much detail, but I'm just going to explain it to you anyway so that you're familiar with the concept. If we look at the genes, you'll see that the nucleotide adenine is going to be replaced by thymine in someone who's got sickle cell anemia. So the triplet code is changed from GAG to GTG. This means that the triplet code on the template strand is changed from TC to CAC. I want you to quickly write out what the mRNA is going to be. The mRNA is going to be GUG instead of GAG. Now, if you've got that table of DNA triplets versus amino acids, have a look at what amino acids this codes for. You'll have valine coded for instead of glutamine. So this small difference in the amino acid sequence doesn't really affect hemoglobin when it's carrying oxygen. It's when hemoglobin isn't carrying oxygen that the hemoglobin becomes less soluble. The molecules form fibers inside the cell and they stick to each other. So the red blood cells are going to be pulled out of shape, giving them that sickle cell shape. The sickle cells cannot carry oxygen, so less oxygen means less respiration. These sickle cells may block capillaries because their diameter is greater than that of the capillaries. This means that you've got an increased risk of thrombosis. And symptoms include tired, listless, and also you have a shortened lifespan. So these are important points to note down for sickle cell anemia. Okay, then we're also going to have a look at phenylketonuria. So people with phenylketonuria lack an enzyme that results in a buildup of phenylalanine in their blood. So this is because of a gene mutation. If this is untreated, it's going to result in brain damage. Okay, let's look at the details of it. You've got phenylalanine, and this is an amino acid found in many foods. Now, normally phenylalanine is converted to tyrosine and then onto melanin, which is the pigment in your skin and hair. And the important enzyme that does this is phenylalanine hydroxylase. What do you think is going to happen if there's a mutation in the DNA that makes this enzyme? Phenylalanine will be built up in the blood and tissue fluid because phenylalanine hydroxylase is not going to be made. And this will result in severe brain damage in young children. And if it's untreated, mental retardation will result. All babies are tested for PKU or phenylketonuria at birth. Their phenylalanine levels are tested. And PKU or phenylketonuria can be prevented if the children are put on a diet that does not contain phenylalanine. Okay, in summary, what have we looked at? Well, what's a mutation? This is when there's a spontaneous change to the genetic material. What are the environmental factors that increase mutations? You've got ionizing radiation, ultraviolet radiation, and chemical mutagens. Do you remember what a gene mutation is? This is a change in the DNA sequence that can affect the amino acid sequence. Can you remember what the different gene mutations are? We've looked at addition, deletion, and substitution, and I also showed you duplication and inversion. What's a frameshift? Well, addition, deletion, and duplication can result in a frameshift, where the change in the DNA sequence will change how the triplet codes are read, and the amino acid sequence will also be changed. Okay, what is sickle cell anemia? It's when there's a base substitution so that valine is coded for instead of glutamine at amino acid 6. So this will result in a change in the beta chain of the hemoglobin, making it less soluble when it's not bound to oxygen. So the resulting blood cells will be sickle cell shaped. And then finally, what is phenylketonuria? This is when there's a change in the DNA sequence so that the enzyme that converts phenylalanine to tyrosine is not made. So phenylalanine levels will be high, and this can result in brain damage if it's not treated. And that concludes our lesson, the end.